Okay, guys, I think we'll get started. Imagine this neuron when it's activated, you feel fear. It happens to be, that this type of neuron, some of you guys know, would likely be in the amygdala, in a particular area of the amygdala, the central amygdala, where if those neurons are activated, you feel fear. If you have a loss of neurons, some people are born, very few, but there are some that are born with the loss of neurons in the amygdala, and they don't seem to be afraid of almost anything. They could pick up a snake, not knowing whether it's dangerous or not, and think it's interesting to look at. So imagine then that this neuron, when it's activated, gives rise to fear, and imagine that there's certain types of events that you encounter that are necessarily going to activate this fear system. A ridiculous being up on a ledge, experiencing or expecting to experience physical violence, expecting to experience loss of affection, things like that. There's certain things that we're innately born to respond to by being afraid. And so what you can imagine then is that there are input neurons to the amygdala that have strong synapses. That is to say, a large number of AMPA receptors. These, which, as we've been doing so far, will continue to represent the AMPA in purple and the NMDA in red. So imagine for a rat, if he's getting a shock to the floor and it hurts his feet, he's going to activate the fear areas, and you'll be able to tell how scared he is, maybe because he freezes in anticipation of that event. But then if he gets a tone before the shock, the tone being a conditioned stimulus, and the tone tells him shock is coming, well, to begin with, the tone wasn't something he was born to be afraid of. And so you wouldn't be surprised to find that this conditioned stimulus, the stimulus that's not innately frightening, inputs to the fear neuron at a synapse that's weak. And that is, in this case, it's as weak as it can get. It's got no AMPA receptors at all. Well, in real life, instead of tone shock, it could be some other, innately frightening event as the unconditioned stimulus. And the conditioned stimulus, instead of a tone, for us could be a particular person, maybe for whom we expect physical violence, or maybe it's a place where we expect something bad to occur, or a situation. So the question is, how do these conditioned stimuli acquire the ability to activate the fear system just as if they were unconditioned, innately frightening or threatening stimuli? And the answer is it's going to be very much like what we've seen so far. Shuya, yeah.
So last time you said the glutamate at synapse only lasts like a few seconds, but what if the tone play and then you wait till like 1 minute and then here comes the shock? Would it still be associated?
Good question. You're right. So that as you increase the amount of time between the conditioned stimulus, say the tone, and the unconditioned stimulus, the learning is less effective. So you might, if you increase the amount of time long enough, you might not learn it at all. If you increase it somewhat, you might learn it less than if the two occurred real quickly, one after the other. So that's a very good question. So yeah, so let's imagine, you've got, we'll use the example of a rat who gets a shock that's always preceded by a tone, and he learns to be afraid of the tone, just as he's afraid of the shock. So imagine to begin with that he hears a tone. Well, he's not afraid of the tone, and that's because this is a weak synapse. The tone can bind to an NMDA receptor, but like in order to depolarize the neuron, you really need plenty of AMPA receptors, which you don't have here. So this explains why most rats, when they hear a tone, they don't freak out. And hopefully, you guys don't either. Now, if you imagine a tone occurring at the same time or just before a shock, and now you say, well, the tone wasn't able to produce any plasticity, wasn't able to activate the neuron, and it couldn't produce plasticity because, again, in order to engage the NMDA receptor, which we were representing again in red, what are the two ingredients that you need in order to activate any particular NMDA receptor within a particular synapse? Why don't you guys chat it in? Exactly right, Michelle. You need glutamate to bind. So you could see that when the tone came on, you actually were able, exactly right, you actually were able to satisfy that first criteria. You got glutamate binding to the NMDA receptor, but that was only one of the two. The second criteria is this has to happen while the neuron's depolarized. Can't depolarize based upon this synapse. There's no AMPA receptors. But if this synapse depolarizes the neuron and that depolarization spreads, and as Shuyi said, it's got to happen quickly while this glutamate is still sitting at that NMDA receptor. But if the two occur close in time, and this multicolored pretty thing represents the depolarization produced by the synapse, you'll now have both criteria satisfied: glutamate binding to the NMDA receptor while the neuron's depolarized. And then, sure enough, you get a new AMPA receptor. You know, maybe that's still not enough for the tone to activate the fear neuron. But if it happens again, where these two, where these receptors are occupied, it's not enough to depolarize the neuron. But if it occurs quickly in succession with the shock input, then that gives you your depolarization. You've satisfied both criteria. And again, you're going to have now even more AMPA receptors. I'm assuming that there's a limit to how many AMPA receptors you get at the synapse. So now it may be that just hearing the tone, or whatever the threatening event was, the conditioned stimulus, now becomes capable of activating fear all by itself. That's pretty much the same idea that we talked about with the concept neuron, and it's the same idea that we talked about just in general about plasticity at NMDA receptors. I want to show you a little animation about the AMPA and NMDA receptor before we leave this.
It binds to both AMPA and NMDA receptors. Although both NMDA and AMPA receptors are permeable to sodium and potassium ions, weak stimulation normally activates only the AMPA receptors, resulting in a slight depolarization of the postsynaptic neuron. When glutamate binds to the NMDA receptor at slightly depolarized or resting membrane voltages, very few ions flow through the channel. This low conductance occurs because the pore of the channel is blocked by magnesium ions, which prevents other ions from passing freely through the channel. Under such conditions, the EPSP will be mediated entirely by the AMPA receptors. Given a stimulus of sufficient strength or frequency, AMPA receptors can depolarize the membrane sufficiently to expel the magnesium from the NMDA channel.
So you guys, notice that he's giving an example where you have so much glutamate being released from neuron 1, say from the presynaptic terminal, that you're able to actually get enough depolarization through the AMPA receptor in order to liberate magnesium at the NMDA receptor. In the examples that we've been talking about mostly, or I think in all of them really, the way we got depolarization was by another neighboring synapse or another synapse on the same neuron where you had a lot of AMPA receptors, and that depolarization spread. We're giving the example more of what would happen during learning. So it's the same principle, but here he's showing you, you could, at least in principle, get depolarization enough to release magnesium from the NMDA, if you just bombard the postsynaptic neuron with glutamate.
The NMDA channel now actively responds to glutamate, admitting not only sodium ions, but large amounts of calcium as well. The calcium acts as an important second messenger, activating several intracellular signaling cascades. Effects.
Okay, so just to point out that the part of this that I'd like you guys to focus on, that we've talked about so far, is simply the idea that in this depolarized neuron, when calcium enters through the NMDA receptor, it activates second messengers, and what we focused on is the idea that some of those actually go into the nucleus of the cell to produce more AMPA receptors.
As a result of the increase in the number of AMPA receptors, the response to a stimulus of a given strength will be stronger than it was before the NMDA receptors were activated. In this regard, the synapse is said to be enhanced, and this physiological change is thought to be one of the mechanisms underlying the expression of long-term potentiation, or LTP.
He describes in that little video clip 3 different ways that you could, he goes through it quickly, but 3 ways you can strengthen a synapse. We focused on one of them. The 3 ways, just for your interest, are increase in the number of AMPA receptors. That's what we've talked about. An increase in the sensitivity of AMPA receptors. We haven't talked about it, and it's not necessary for our purposes. And an increase in the likelihood of release of glutamate from the presynaptic neuron. So now we can talk about something pleasant. As opposed to fear, we can talk about reward learning. So let me just say hi to you guys for a second. All right, so let's recall that learning to associate one stimulus with another is called Pavlovian or classical conditioning. So here's a simple animal example. A rat learns to associate certain things with a reward, like food. A person may learn to associate certain things with drugs and other rewards. So let's say for the rat, he learns that a certain tone always precedes him getting his favorite food or getting his daily food. The conditioned stimulus predicts the unconditioned stimulus. He hears the tone, he gets all excited because he knows that means that food's coming. Notice that the conditioned stimulus always, it's the conditioned stimulus that predicts the unconditioned stimulus. It's not the food that predicts the tone, it's the tone that predicts the food. That's the way this type of learning goes. All right, so you always put like the most important thing, in this case the food, at the end. And the idea is you're trying to learn what is it in this world that tells me, oh, here comes that good thing. All right. Also notice that, you know, the food was good before you did any learning trials. You know, you were born liking the food. But the value of the conditioned stimulus, I mean, the tone had no value to begin with. It was just some sound like any other sound. It's because the tone, the conditioned stimulus, predicts this good thing that the tone takes on a positive value. Before learning, the tone was just a neutral sound. The conditioned stimulus was just a neutral stimulus. All right. And again, notice that that's not true of the unconditioned stimulus. Unconditioned means unlearned. You didn't need to learn it. The unconditioned stimulus, food, say, doesn't require learning, doesn't require conditioning. Conditioning is just learning. You don't need to learn that the food is valuable. It's hardwired. It comes from the way your brain circuits work. You know, you inherited genes that created brain circuits to enjoy food. You know, otherwise you'd be in trouble. So, you know, drug addiction, part of the thing that makes it so difficult to help people with drug addictions is that it's tapping into these reward circuits deep within the brain that evolved, you know, to keep us alive, you know, finding the important rewards in our environment, like food. So let's say in this case, the person, instead of learning about some situation that predicts food, they're learning about a situation that predicts this other reward, an artificial reward, say heroin. All right. Maybe the conditioned stimulus is the street corner that predicts reward. Or maybe it's also, maybe it's not just a street corner. It's a street corner with a certain guy standing on it that tells you that the heroin's available. Again, the conditioned stimulus precedes the heroin, and the value of the conditioned stimulus is basically nothing. It's just a street corner until that street corner becomes associated with the heroin. Then this is not just a street corner like any other. This becomes like a fantastic street corner, you know, if you're really craving the heroin. Not true with heroin. You don't need any learning for it to have value. Its value comes from the fact that this drug is activating neurons in the brain that evolved for all types of other rewards, natural rewards. But this heroin is activating these neurons to an extent that evolution never really expected to be possible. And so you're getting this extreme reward response, you know, maybe good for the moment. There's obviously a lot of danger and negative consequences to it. But it certainly drives the reward system. And it makes you very likely to learn what stimuli in the environment are predictive of that reward. Now, let's say Tom has a heroin addiction, and he goes to a clinic where he's abstinent. He doesn't take any heroin for 2 months. So he may have been on heroin for years. Now he goes in, he doesn't take it for 2 months. You know, he's on a certain floor of this hospital. When he leaves, he no longer has withdrawal symptoms. He no longer has drug cravings. However, he gets home, he's driving around, and suddenly he has intense cravings. Like, somebody raise your hand. What happened? He saw the same corner where he would buy drugs. Exactly. You know? So, I mean, in the hospital, it's fine. The physical craving's gone, but the conditioned stimuli that have become associated with the drugs and the drug reward, they're still out there. They haven't lost their value. You know, it may be during years that street corner acquired this strong association to heroin. And, you know, seeing that street corner gives you this great urge to have heroin. Even when you're no longer having withdrawal symptoms, you see that street corner and you're right back into it, you know. So this is one of the real problems with treating drug addiction. Now, if he's repeatedly, yeah, Jade, so that's what I'm going to get to now. If he's repeatedly exposed to the conditioned stimulus without the heroin, it may be that he weakens his association between the conditioned stimulus and the heroin. And the name for that type of learning, as I think a lot of you guys know, is extinction. So you say, you know, he got the CS, the conditioned stimulus, in the absence of the US, in the absence of heroin, and you call that, you say that he went through an extinction procedure, or he went through extinction. Now, let's say that Tom learns a new way to inject heroin that produces a particularly intense high. You know, I don't know how he did it before, but I don't know. He's got sort of a new type of, I don't know, place on his arm where he injects himself, or he injects himself more forcefully, presses the syringe more quickly, whatever it is, he gets like the best high he ever had. Before we go on, who remembers what's different about operant learning compared to classical conditioning? What's operant learning about? Operant learning is when you associate something important, like it could be heroin, whatever, not with some other stimulus like a street corner or a tone or a light, but with something you do. For example, a new injection procedure. It's something where your behavior makes a difference in terms of when you get the reward. Well, so now we're not talking about classical conditioning. We're now talking about something to do with your behavior and the reward. That makes it operant because your behavior is part of the equation. I'm not speaking just so that you can read this yourself. The behavior is called your operant behavior, this new injection method. The reward is the reinforcer. It's the thing that makes you want to do that behavior more frequently in the future. Let's think about some real-life examples of a CS-US association, and then we'll deal with operant behavior. So remember, the CS and US need to both be stimuli things. Neither one can be an action. The US can be rewarding or aversive. So why don't you guys type in just some examples of a conditioned stimulus that can be associated with an unconditioned stimulus. When I lived with my parents, my dad would always cook dinner, and I would get hungry when I could hear him taking pans out of the cupboard. Yeah. So the conditioned stimulus is going to be hearing the sounds, hearing him do the stuff he did with the pans. And the unconditioned stimulus was dinner, which occurred soon after, and they would get associated. So now how about reinforcement? So with reinforcement, we're talking about operant behavior. Well, that's good, Michelle. Studying to get good grades, so that your brain has noticed that the more you study, say, the better the grades. And so there, instead of just some conditioned stimulus environment getting paired with some outcome, it's your own behavior, studying to get good grades. Studying, let's say, and the grades are the reinforcer. Very good. Let me get a couple more examples of operant behavior. That is to say, reinforcement. Yeah, golden star sticker is the reward, and good behavior is the operant. So you do a good behavior and you get the star, and presumably that star, that reward, is rewarding because you've associated with approval or whatever, but it absolutely counts as a reward. The more you work out, the more built you get. Yeah, you're noticing again a relationship between your behavior and an outcome. For example, in the gym. Let's look at the brain. Animals will press a lever not just to get things like food reward, but also get electrical stimulation of certain brain areas. They're especially motivated to receive stimulation of a group of neurons in the brain called the medial forebrain bundle. When this was discovered, it was like a headline in all. It was discovered in Canada, actually, by some researchers in Montreal. But it hit the U.S. papers, all over the world. These headlines, pleasure center of the brain discovered. Now, since then, we know more about it, and it's not just the medial forebrain bundle, but this was kind of the first understanding that there's parts of the brain that are dedicated to the reward experience. These rats, they would press for this electrical stimulation, which basically just means they're artificially activating these neurons in this medial forebrain bundle. It's a group of neurons. And here you see in the human brain a colored outline of the medial forebrain bundle, and you can see it goes from lower regions of the brain, like the brain stem, the midbrain, all the way up until to the more dorsal or higher parts of the brain, including the cortex, and we'll see some other important areas in between the cortex and the brainstem. But these rats, they just kept pressing, and they never got tired of it. Like, it's not like food where they would get satiated. And in fact, once a rat learned that this lever produced stimulation of the medial forebrain bundle, you would have to turn it off so that it would eat, because it was didn't matter how long it had been since it last ate. It just wanted to press that lever for we call it MFB, medial forebrain bundle, for that MFB stimulation. So you can see why, if you're interested in understanding about reward in the brain, and especially if you're interested in understanding what's going on with addictive behavior in humans, this is a pretty exciting clue. It makes you think, God, if we could understand how this works, we may really get somewhere, So what accounts for the powerfully reinforcing effects of medial forebrain bundle stimulation? So it's not that obvious, because the MFB, the medial forebrain bundle, contains a lot of different types of neurons. Glutamate neurons, GABA neurons, dopamine neurons, norepinephrine neurons, some serotonin neurons. So you can't really tell which neurons in particular are responsible for this reward effect. You just know it's something in the, like, think of the MFB as like a big, thick cable of wires going to various different places. You don't know which of the wires, or maybe it was all of them, but maybe it's just a couple of them that are really the key to the reward effect. And the question then became, is there a way to figure out which ones are key? And, you know, one question is, could it be dopamine? Now, why did people jump to dopamine? Well, one reason they jumped to dopamine right away as a kind of a likely suspect was because it's already been known that drugs like cocaine and amphetamine that have addictive potential, you know, people will do those, you know, work or self-administer for those types of drugs compulsively, just like these rats would lever press for the MFB stimulation. You know, could it be that this MFB stimulation is activating dopamine? Well, sure enough, it contains a lot of dopamine neurons, this MFB. Maybe they're the ones that are responsible for the reward. So they did some experiments where they would have a rat pressing a lever, and it would get electrical, stimulation of the MFB. And they said, what would happen if this rat or this group of rats that are pressing and they get this little yellow thing going into the brain is supposed to be the electrode that's stimulating neurons in the MFB. You know, what if we give a drug that blocks dopamine? So in other words, this flat line, say, are the animals that know how to press the lever to get MFB stimulation, and they're not getting any drug. So they're just pressing boom, 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 boom. All right. And then what you say is, okay, once they've learned that, let's take another group. One group gets to keep doing that, but the other group, we're going to give them a drug that blocks dopamine receptors. Is the MFB still rewarding? And what they found is that these animals under the influence of the drug, and the drug took complete effect by minute 1, but they still pressed a lot, which actually was good because it showed that the drug's not sedating them, because otherwise that could be the whole explanation. But what happened is pretty soon they gave up pressing as if the MFB lost its value, the MFB stimulation lost its value when dopamine receptors were blocked. Okay, so lever presses per minute is in the y-axis. Lower means fewer lever presses. Time is on the x-axis. As time goes on, the animals, red is the animals that got the drug that blocks dopamine receptors. The animals whose dopamine receptors were blocked lost their interest, apparently, in pressing the lever for MFB stimulation, suggesting that maybe it's the dopamine that's responsible for the reward. All right. So I'm going to give you this one more slide before we quit for today. Within this MFB are these dopamine neurons. So I would like you guys to be familiar with the dopamine system. There's really 2 places in the brain that have the dopamine cell bodies. They're these little clusters in the midbrain. So it's not in the spinal cord, it's not in the brainstem, it's not in the pons or medulla or pons. It's in the midbrain. So it's still pretty low in the brain, but it's not as low as all that. Basically, if you went from the bridge of your nose, say, straight back, you would find these clusters of dopamine cell bodies. You know, maybe in the human brain, these whole clusters would be maybe the size of a penny or something, you know. I mean, and you have this whole big brain, so they take up a small area. But these cell bodies give rise to axons, as you can imagine. And those that are in the, in red here, I'm showing you an area called the ventral tegmental area. That's one cluster of dopamine cell bodies. Okay, and they send their axons to 2 key places. One of them is actually this red line, which goes to the cortex. But the other, which is particularly important we think, for drug addiction, is this little green line. If you follow the green line from the red circle, you see the Vta dopamine neurons are sending their axon terminals to this little orange area. Okay, I'm going to show you what that is in a second. And the substantia nigra is another group of dopamine cell bodies. And they're going to send these purple lines, these axons, to another brain area. And let me show you what they are. I'll do this last thing. Okay, this may seem like a lot at first, but you'll get used to it quickly. Let's start with the SN. That's in purple. The dopamine cell bodies in the substantia nigra, they project mostly to this area, which is called the striatum. Okay? So that's this whole area here. And these are the dopamine neurons that if you look in the brains of Parkinson's patients who have trouble moving, you see they have a loss or a death of these dopamine neurons. But more important perhaps for addiction is if you look at the Vta dopamine neurons, the ones with the cell bodies depicted here in red, they, in addition to projecting to the cortex, they project to this orange area, which is called the nucleus accumbens. All right? So you're going to have to look at this and just get used to it if you haven't ever heard this before. The dopamine neurons, the first group, the motor-related neurons that go from the substantia nigra to the striatum, are called nigrostriatal dopamine neurons, or it's the nigrostriatal dopamine pathway. The ones that start in the VTA, it's just neighboring. You know, the VTA is just more close to the midline, basically. The SN is a little bit further towards the ears, but they're both clustered towards the center of the brain. The Vta dopamine neurons project mostly to this little orange area, the nucleus accumbens, which is called the mesolimbic dopamine pathway. And as you can see, they also go to the frontal cortex. Guys, yeah, so have a wonderful couple of days, and we will see each other on Thursday.
Thank you.
All right, guys. Have fun.
Bye.
Bye. Anybody eating a lot of ramen noodles these days?